Okay. Sorry, I failed to record that for the introduction. I get distracted on that. Anyway, Suzanne's going to present. She has agreed to have questions during the presentation. If you'll populate them in the chat, I will monitor them and invite you to ask your question. And we'll go for about 40 minutes, 35 or 40 minutes. And then there can be questions at the end as well. And thank you, Suzanne, for presenting today. We're really looking forward to it. And we'll turn the time over to you to share your screen.
Okay, sounds good. Okay, firstly, big thanks to Diane for the invitation. I got lucky enough to visit Utah about 6 years ago now. So I'm delighted to be able to present, and I think the virtual format has been really helpful for me, catching up on all the other people's talks. So I'm really delighted to be able to participate and being on the other side of the world. So today I'm going to talk to you a little bit about the role of iron in the lung and some of the things we're doing in the lab. So it's sort of a big overview of everything we're doing. Please interrupt questions or comments. So I'm going to talk to you, just show you a little bit of where we are now. So I'm now based at Trinity College Dublin in Ireland. Trinity College is right in the centre of Dublin City. It was founded in 1592, so it's the oldest university in Ireland, and it is one of the 7 ancient universities of Britain and Ireland. So it's got a really rich history and a really beautiful campus. It also has, we're famous for our long room, which is a 300-year-old library that hosts the Book of Kells, which is an ancient manuscript. And this is one of our top tourist attractions in Dublin when it's open. Due to COVID, it's been closed, but this is just a snippet of what it's like inside. So my lab in Dublin, and also I have a little bit of presence in New York City and Weill Cornell. We're interested in metabolism and lung biology and disease, including iron and the interplay of iron and the mitochondria in lung biology and disease. So just to give you a little overview of iron in the lung. So the lung as an organ and how it relates to iron, we know that the lung is continuously exposed to exogenous sources of inhaled iron. So just like the GI tract, it has a window to the outside. This inhaled iron may contribute to up to 6% of the total iron levels in the lung, which is minimal compared to what the GI tract can get up to. And we believe, but we don't really know, that rest of the iron comes from the pulmonary vasculature. The lung itself would have a lot less iron than the heart or the liver. And we believe that most of the iron is found in tissue, with the rest in bronchoalveolar lavage fluid, which is the fluid lining the airways and the alveoli. We believe that there's also some iron that's unbound in the lavage fluid. Just to give you a brief introduction to some of the cells in the lung today, I'm going to talk to you mainly about the alveoli and cells in the alveoli, but obviously there are numerous different types of epithelial and endothelial cells and various other stem cells, and have very specialized function in the lung. But as I said, today I'm going to talk more about the alveoli. So in the alveolar space, you have an alveolar sac here surrounded by alveolar type 1 epithelial cells, which are responsible for the gaseous exchange function of the lung. And then you have alveolar type 2 epithelial cells, which are highly metabolic. Their main role is to produce surfactants, which reduces the surface tension in the lung and allows for gaseous exchange. Surfactant is composed of 90% lipid, and so hence why these guys are really metabolic. And then you have the presence of alveolar macrophages and sometimes other innate immune cells. Alveolar macrophages there's very few and far between in a healthy lung, but obviously in response to injury and infection, you see an influx of these cells into the alveolar space. So as a lab, we're interested in nutritional immunity, or the process by which an organism sequesters trace metals such as iron or zinc to limit the pathogenicity during infection, so to hide these metals away from an invading pathogen. And actually, the School of Biochemistry and Immunology in Trinity College Dublin is one of the world leaders in immunometabolism research. So the fit here of me coming here and expanding my lab here is amazing, because the resources that they have and the investigators they have here is going to really help me to explore this concept of nutritional immunity and iron in the macrophage. So I'm going to tell you how I got into iron. I started my postdoc in a lab, a very well-known respiratory physician scientist, and he worked on autophagy at the time and didn't work on iron. So how I got involved and how I got into iron. So I need to tell you first about a disease model, or a disease that we focus on in the lab, and what my mentor Augustine focused on when I first arrived in his lab. So chronic obstructive pulmonary disease, for many of you that don't know, it's a highly prevalent disease internationally, and especially in lower and middle income countries. It's thought to contribute to over 3 million deaths a year, and it's the third leading cause of death worldwide. It affects nearly 16 million people in the US and costs the US healthcare systems billions of dollars annually, as many of these patients require consistent or constant hospital admission. We believe that chronic obstructive pulmonary disease, in oversimplification, consists of two major phenotypes or clinical diagnoses. One is chronic bronchitis, which involves the excess production of mucus in the airways and inflammation. The second is emphysema, which is the loss of alveolar epithelial cells, leading to less surface area for gaseous exchange. It is a smoking-related lung disease. However, only 20% of smokers develop COPD, and there are quite a significant number of patients who have never smoked. So for many years, it is well known that there may be a genetic risk factor for COPD, or multiple genetic risk factors for COPD. And so this is how I got involved in iron. So when I arrived to do my postdoc, Augustine and Don DeMeo and Ed Silverman at the Channing Institute had just published a paper showing that IRP2 is a genetic risk factor for COPD. So they found a number of SNPs in the IRP2 sequence that confer susceptibility in a familial-based COPD cohort. And they looked in detail at these SNPs and also found that IRP2, which I don't know if many of you know this, but IRP2 is just upstream of 3 nicotinic acetylcholine receptors, meaning that IRP2 may have share some co-regulatory factors with these guys, and perhaps why IRP2 may be sensitive to smoking, as this is a smoking-related lung disease. So I really don't have to tell this audience what IRP2 is. But it is a master iron regulator in cells. It can positively and negatively regulate the translation of many proteins involved in iron metabolism, and is responsive to low and high iron levels. So Dawn showed in her paper that IRP2 levels were much higher in the tissue of COPD patients at the protein and RNA level. And we subsequently went on to show that in a marine model of COPD, which involves exposing mice to cigarette smoke, that IRP2 levels were also increased at the message and at the protein level. And we went on to show that cigarette smoke, we believed, induced mitochondrial dysfunction via iron overload in the mitochondria, and that this was mediated by IRP2, and this contributed to COPD. This obviously was probably only one mechanism of how IRP2 regulated the response to smoke and contributed to dysfunctional phenotype seen in COPD, but it was a nice story. So this led me to start reading and think about, well, okay, what does this mean, iron and COPD, and how real is this, and how prevalent is this a real thing? So we did a big literature review of everything. We found that SNP variants in IRP2 and IL-6 have been known to increase susceptibility to COPD. HFE mutations, surprisingly, also have, as have porphyrias, and ferrous metallurgy workers are known to have a higher susceptibility to COPD, as well as other chronic lung diseases. But there is a lot of evidence to suggest that if your iron levels are misregulated, that you may have or be more prone to getting this lung disease, especially in the context of smoking. So to move my lab on, we decided, okay, where is the iron in these COPD patients? So we know that iron is higher in the COPD patients. IRP2 is higher, it's bringing in more iron. Where is the iron? So if you actually stain a section of lung from a COPD patient with Pearl's Parisian blue, what you'll see is that the predominant staining occurs in alveolar macrophages that infiltrate the lung. So this is quite a severe COPD patient. What you can see is that their macrophages are intensely stained with Pearl stain. And it's this wasn't a new thing. Actually, many other people had shown that the number of iron positive macrophages in a COPD patient correlated with disease severity based on gold scoring, which is a scale that we use to grade COPD severity. The higher, more severe subjects with gold 4 had more iron positive macrophages, and this correlated with the degree of emphysema by CT scan. So there's a lot of nice papers dating back many years to show that there is something there with iron overload in the macrophages. And we also know that macrophages are dysfunctional in COPD. So COPD patients can sometimes have 25 times the number of alveolar macrophages in the airways and alveoli, but they don't work correctly. So they can't clear bacteria very well. They have impaired efferocytosis. They have altered levels of pro-inflammatory and pro-resolving markers. And we believe that this contributes to the immune system disarray that you see in these patients. They have a lot of inflammation, and they also have increased susceptibility to bacterial and viral infections. And so we are trying to see if we can get to the bottom of that a little bit. We also know, and many people have talked about this before in this meeting, but we know that iron metabolism plays a role in macrophage plasticity. This obviously is a diagram, a very oversimplified diagram of macrophage plasticity involving the M1 and M2 macrophages, and others have published that an M1 macrophage is thought to be more iron retaining to engage the native burst, and an M2 macrophage is thought to be more iron recycling to enhance resolution of injury and infection. Again, this is an oversimplification, and I'm sure especially in the lung, there are many other subphenotypes and we've no idea what their iron handling is like, but this was just a background to the iron may play a role in macrophage behavior and plasticity. So we believe that if you could look at iron changes in the macrophage and how then this would then directly affect alveolar macrophage function, and then in turn how this would affect bacterial clearance or polarization or immunometabolic changes in the alveolar macrophages specifically. And the alveolar macrophage itself is quite different to other macrophages found in other tissues because it's quite, it adapts to its niche. It's embedded in surfactant, which we know is enriched in lipids. And so its behavior and its identification, or how it looks, is different to other macrophages that you would see in other compartments. So in the lab, we use two main models of COPD. The first one is smoke-induced COPD, and this is the most common physiologically relevant model. The second is a neutrophil elastase model, which basically mimics emphysema. So the elastase just breaks down all the alveolar structures. But today I'm mostly going to talk about smoke-induced COPD. So some of you have met Bill Zhang in my lab in Cornell. He's an MD. He's writing a KO8 at the moment, so pretty serious about continuing to work on iron in the lung, just like I am. And he has generated some really nice data. And the first thing I'm just going to talk to you about is that how well this model can recapitulate human COPD. So if you expose animals to around 8 to 12 weeks of smoke, what you can see is their bronchoalveolar lavage fluid macrophages, when you spin them down, are clogged full of, they're black. They're just like a smoker's cells. When you put these on flow, you can see that they are bigger and more granular and contain a lot of crap. They're actually really hard to work with when it comes to fluorescence because they're quite autofluorescent, but this just gives you an idea of how well the model works. When you cytospin these cells and stain for iron, what you see is there's quite a heterogeneous staining pattern of these macrophages in response to smoke. So you can see not all of the macrophages stain for pearls, and some of them are brown and some of them are intensely blue. There's punctate structures, and we don't know what any of this is, but it's really interesting. So we think this also led us to kind of think, we really have to go and find out what's going on. He has also shown that when you look at iron, some iron markers such as transferrin receptor or ferritin heavy chain, they're increased upon smoke in these alveolar macrophages. And in vitro, we can give essentially liquid smoke to alveolar macrophages and mimic the same effects in vitro. So we have a nice in vitro model to work with. We've carried out bulk RNA-seq of macrophages isolated from smoked mice. And what we see is there is an enrichment in transition metal ion homeostasis. And if you zoom in on this by IPA analysis, what you'll see is there are a number of proteins that change upon smoke in the alveolar macrophages. Now, we used I think it was an 8-week model for this smoke exposure, and so it could be considered quite mild. I'm sure if we smoked even for longer, we'd see stronger effects in some of these proteins. We've also published that hepcidin and ferroportin are differently expressed in human tissue-derived lung macrophages in individuals with COPD and in smokers, ferroportin being increased dramatically by smoke, as you can see here. When we try to mimic this in our in vivo smoke model, you'll see the ferroportin levels increase up to 8 weeks of cigarette smoke exposure. We do not really get the same effects with hepcidin. It's very minimal. It's sort of not surprising, but we were interested in looking to see how smoke affected the normal iron response of the macrophage to infection. So we know that hepcidin is essential, an innate response to the macrophage seeing infection, and you see downregulation of ferroportin, so the macrophage tries to keep all its iron inside. And so we wanted to see if smoke could affect this phenomenon, and what we were able to show is that if you take alveolar macrophages, expose them to LPS, you see increased hepcidin here, as you would expect. But if you pre-incubate these macrophages with cigarette smoke extract before you give LPS, you see a blunted increase in hepcidin, or an inhibited response to LPS. Same with ferroportin levels as well. I don't show that here. You see enhanced ferroportin. So we then decided to explore the fact whether loss of hepcidin globally could affect the injurious response of the lung to smoke, and it didn't. So we spent a lot of time and effort doing these experiments. We found that the hepcidin deficient animals had similar numbers of infiltrating leukocytes into the lung lavage fluid. We found that they'd similar levels of injury by BALF protein levels and BALF IgM levels. What was interesting is ex vivo, when we took alveolar macrophages from our hepcidin deficient animals and exposed them to Streptococcus pneumonia, which is a common pathogen that that COPD patients are more susceptible to. What we found is that the hepcidin animals seem to have a greater bacterial burden, and that the hepcidin deficient macrophages could not respond in terms of producing IL-6 or TNF alpha when they were exposed to Streptococcus. So this would suggest that loss of hepcidin has some sort of effect on the response of the lung to infection. We tried to do an in vivo experiment where we just chose 2 doses of Streptococcus pneumoniae and gave it to our hepcidin deficient mice. We saw mild effects where they may have a more impaired response to strep. We would have loved to have been able to do a greater dose and time course of this and really tease this out, but unfortunately resources and breeding, and this was all, we couldn't have done it all. So this is, we think there's something there, and it was interesting observation, and we'd like to explore it a little bit further. So one of the priorities of my lab is to really understand what we call Pmax. So these are iron-loaded macrophages in the lung, and this is, a really nice concept that we really learned a lot from Dr. Alyssa Hasty on her recent publications, that there may be a heterogeneous population of macrophages in the lung, and one of those subpopulations may be a macrophage that's more prone to iron recycling or retention or release. So we're interested in what we call these Pmax do in the lung. How do they accumulate iron in COPD? A lot of people will just think, okay, well, there's iron in the cigarette smoke, and that's what's causing the increased levels of iron in these macrophages. But we've measured iron in cigarette smoke, and it's not that significant. We've tried removing the iron from cigarette smoke extract and giving through Chelax, and we don't see too much differences in the responses that we get. Some people have asked us, is there any pulmonary hemorrhage? And, is there red blood cells floating around that maybe contribute to this? But classically, and I'm not a physician, but I've worked and spoken with a lot of physicians, that classically a hemorrhage wouldn't be a massive thing, especially not in early COPD, was where you see this more increased susceptibility to infection and where you see these iron macrophages. So we don't think maybe it's partly to blame, but we don't think it's everything. So we're interested to know, how do they accumulate iron in the COPD lung? What is their biological significance of iron accumulation? Does it affect their phagocytic ability, which I know others have shown it can? Can it affect their immunometabolism, efferocytosis, all of these different functions of macrophages? We're really interested in exploring how iron regulates those. And what's the clinical significance of iron accumulation? So this is something Bill is very interested in, and figuring out if patients have more iron, Fmax, are they a different type of endotype of COPD patient, for example, and should we be treating them differently to other COPD patients who don't have the same level of iron overload? So one thing Bill has tried to do, and this is again adapted from Dr. Hasty's work, was to take bowel macrophages and try and sort them based on their ferromagnetic attributes. And unfortunately, we couldn't get this to work a couple of times we tried, and we just think that the starting material from a lavaged mouse is much, much smaller than it would be from adipose tissue. But we this concept is something we keep going back to, and we'd love to be able to sort these macrophages. So we're exploring other markers such as transferrin receptor or other things to be able to tease these populations out a little bit more and study them functionally and see how they differ from the other populations in the lung. And so one of Bill's main projects, and this is the focus of his KO8, is to understand iron metabolism in the macrophage, specifically NCOA4 and ferritin turnover in the alveolar macrophage in the COPD lung. And we hope that we have a story for you next year, or maybe even earlier, that we can get some feedback on. But there's some exciting data, and Joe Mancias has given us mice and everything. We'd love to talk about this in the future, but this is Bill's main project, and so watch out for him. So another aspect of my talk is, okay, so we know that iron levels are higher in the macrophages of COPD patients, but what about the microenvironment of the alveoli or the alveolar space in these patients? Again, others had shown that iron levels were higher in the lavage fluid, but very low numbers, and clinically we didn't, they never really explored what this means. So we went to a database in the US called the Spiromics cohort, which is a multi-center US based, one of the largest longitudinal COPD based cohorts in the country. And we were able to obtain bronchoalveolar lavage fluid from these patients, and we measured iron and ferritin and lipocalin, and we wanted to see if the iron and ferritin levels changed in these patients. What we found was that in smokers and COPD patients, the iron levels were higher than in the controls. And you can see this pretty tight with both iron and ferritin. Lipocalin also behaves the same. I don't have that data here. What we found was this local lung ferritin and iron levels did not correlate with systemic markers of iron storage or inflammation. So we can see here on the left that bowel ferritin did not correlate with plasma ferritin in the same patients. And you can see it did not correlate with hemoglobin levels or CRP. Neither did bowel iron levels. And as you can see here as a control, we see that log plasma ferritin did correlate with hemoglobin and CRP in these same patients. So we think that local lung iron overload is a distinctive and separate phenomenon, and we want to explore that again a bit further. Something that we also showed in this publication was that patients who have more exacerbations or an event requiring medical intervention, those patients have higher bulk iron levels and significantly higher levels, which meaning that the more iron that's there, maybe the more likely they are to have more respiratory infection, as exacerbations are usually associated with an underlying respiratory infection. And so we were lucky enough to have 16S sequencing from the same patients in the study, and we'd started doing preliminary analysis. Again, hopefully have this story out in a little while to show that iron levels in the lavage fluid associate with certain types or phyla of bacteria. And again, this will provide us with more information moving forward of how to study this, the role of iron in the lavage fluid in more detail. So in Dublin, I have a really talented microbiologist, Claire Healy, who came from Weill Cornell Medicine. She's interested in looking at non-tuberculosis mycobacterial infection in the COPD lung and how iron plays a role in this. So that's something that we're pursuing here. And then I have a graduate student, Lynn Faherty, who's also working on how iron in macrophages will tate the response of the macrophage to infection, and how the two fight for iron. So In terms of COPD, we know, just to summarize, that IRP2 regulates mitochondrial iron in marine smoke models. We know that local lung iron overload associates with a disease exacerbation in COPD-6. We know that alveolar macrophages accumulate iron, which may contribute to the dysfunction, and that alleviating iron in the lung may offer therapeutic potential. So I didn't show some data, but back in 2016, we showed that if you give deferriprone to mice and expose them to smoke, they're protected from the effects of smoke in terms of inflammatory responses. So we are trying to explore if that's a feasible therapeutic in the lung or not. So one other thing we're interested in is iron availability in the alveolar epithelial cell. So you'll see here, again, this is just a schematic of the alveoli. We're interested in how iron plays a role in the function of the alveolar epithelial cell, especially the type 2 epithelial cell, which I mentioned produces surfactants. So we're interested in how iron plays a role in the repair and regeneration of the lung and in the response of the alveolar type 2 cell to injury. We're also interested in the role of iron in senescence phenotypes that you would see with in lung disease. So we have some nice data there that iron may play a role in senescence in COPD and in other chronic lung diseases. And so a talented fellow in my lab, she's now back in China, is Jilin Huang. She was here for 2 years, worked on this project and really started and created the base for some of the work that Niamh Williams, a postdoc in my lab, and Sarah Kenny, a graduate student, are for their projects looking at the role of iron in alveolar epithelial cell dysfunction. So one thing I wanted to talk about today is how we maybe could study iron in the alveolar epithelium and what we can do to manipulate iron. And so we started looking at the role of bleomycin, which is normally a chemotherapeutic drug, but it's known to exert its chemotherapeutic effects by intercalating iron, and it utilizes iron to oxidize DNA and cause damage in cells. And we know that if you give bleomycin to a mouse, they develop fibrosis after about 21 days. They get severe type 2 damage, they get some inflammation, and then they get fibrosis. And many people use this model as a model of idiopathic pulmonary fibrosis, which is another chronic lung disease of interest. We had published a paper in 2019 that had carried out RNA sequencing of type 2 alveolar epithelial cells from this model, where bleomycin treated model, and we saw that there were changes in iron transport and many other iron related pathways, which is not surprising given bleomycin is such an iron effector. So we said, okay, can we use bleomycin as a way to manipulate iron in the lung? And so many people have actually given bleomycin to mice and looked at iron levels. And so we did the same, and we found that non-heme iron levels increased after bleomycin treatment. And here on the right is a number of recent papers, all in the last 3 years, who have shown the same thing, that iron levels increase. And this is not surprising, as you would see after 14 days, quite a lot of hemorrhage in the lung after bleomycin treatment. But what we decided to do is we decided to give bleomycin and then isolate alveolar type 2 cells after 8 days and measure iron levels. Because the idea being that if bleomycin's mechanism of action is to move iron or to use the iron to create damage, then maybe it's sucking iron out of places it shouldn't be. And so we thought that the alveolar type 2 cell may be a target of this. And what we found is if you isolate alveolar type 2 cells from these mice and measure iron levels by graphite furnace AAS, you see lower iron levels in these cells. Now, just as an offside or a note, as I said, there have been a couple of papers that have looked at the role of iron in the bleomycin model. Some of them have shown that alleviating iron is good, and some have shown that iron has no effect on bleomycin. So there is a little bit of conflict in the literature about it, but I think that's mainly due to the fact that some people gave iron or manipulated the iron before bleomycin, others gave it after. And definitely there is a lot of hemorrhage in this model again, so you're kind of messing around with different things. But they're worth reading these papers to really tease out what's going on in the pulmonary fibrosis world in terms of iron. But we wanted to use bleomycin as a way to deplete iron in the alveolar epithelium. And as I said before, we're really interested in mitochondrial iron and mitochondrial iron transport. So the most obvious transporters are the only identified transporters of iron into the mitochondria, mitoferrin 1 and mitoferrin 2. So we looked at the expression of mitoferrin 1 and mitoferrin 2 in isolated alveolar type 2 cells in the bleomycin model, and they were lower in response to bleomycin in the type 2 cell. And thanks to Diane, who gave us some flocks mitoferrin 1 and 2 animals, we were able to cross these animals with a type 2 alveolar epithelial cell specific Cree system. So it's an inducible Cree that deletes the mitoferrins in the type 2 cells. And what we did is when you induce these, the loss of mitoferrins with tamoxifen, what you see is after about 4 to 6 weeks, you see that about 60% of the mice die. And what we started to notice was that there was severe neutrophilic pneumonia. You see increases in neutrophils in the lavage fluid, and the number of lavage cells dramatically increases. So if you look in detail at their lungs, they're pretty sick. We think they have interstitial pneumonia. And this here, iron levels are just to confirm that the type 2 cells in these mice actually have less iron, and they do. So we're interested in learning why this would be the case. What are the mechanisms why this is happening? And we've got some really nice data to follow on from with on this. It's sort of exciting. One thing we've also done is we've created an in vitro model for deleting mitoferrin 1 and 2 using CRISPR-I, or not deleting completely, but reducing the levels of mitoferrins in the alveolar 2 cells in culture. And what we see is when we delete mitoferrin 1 and mitoferrin 2, we see that the cells they're viable, but there's a lot of death after we delete them. When we delete mitoferrin 1 or 2 using CRISPR-I, we see the total iron levels decline. We see that mitochondrial iron levels decline. So the way we do this here is we create mitoplasts. So we create mitochondria with no outer mitochondrial membrane, and then we measure iron inside those by graphite furnace AS. And we see levels of mitoplast iron decline in the mitoferrin 1 and 2 CRISPR-I cells. We see that aconitase activity declines. This is just whole cell aconitase, it's not mitochondrial specific. We see heme levels decline. And we also tried this mitoferrogreen that we'd seen in the literature and shown that the intensity or fluorescence intensity of mitoferrogreen also declined. Just to disclose, we did have to load these cells with a lot of FAC before we could see any of this fluorescence. So we managed to see a difference between the CRISPR-I cells and the controls. So we've got a nice in vitro model to really tease out some of these mechanisms further as to why the mitoferrins would be important, and specifically use them as a way to manipulate iron in a type 2 cell. So what we're really, as I said, interested in is bringing all of this together is in the lung or in the alveoli, will an iron macrophage modulate alveolar type 2 cell iron levels as well, and then how this leads to inflammation or infiltrating monocytes or neutrophils, and how this helps or hinders the lung repair after injury. And so these are a lot of open questions. And again, this whole idea of looking at the interaction between these cells is, really important in my mind as to what we should be doing next. So just as for the last few minutes, I just wanted to talk 2 slides on something else we're doing. And again, this is spearheaded by Bill Zhang, and I was in New York at the beginning of the COVID pandemic, and Bill is a pulmonary critical care physician, so he was on the front lines. And so something we felt we needed to do at the beginning was to start looking at this hyperferritinemia thing and see what we could do to add to the story. And so we've just got some really nice work on the role of macrophage ferritin in COVID-19 induced acute respiratory distress syndrome. And so we were able to go to our New York Presbyterian cohort, and this obviously has been shown many, many times, that the more ferritin a patient or individual has upon admission, the more likely they are to die in their hospital stay based on their ferritin levels. And again, whatever their maximum ferritin levels have reached, and the highest amount, if you compare those to survivors and non-survivors who've been intubated, you'll see there's a much higher level in those that did not survive. We've looked at ferritin many different ways in this cohort. We've looked at baseline ferritin upon admission and delta ferritin between the baseline and the maximum. And so we've got some really nice ways to represent this data. And then mechanistically, we were interested in seeing what form the ferritin is in COVID-19. And we think we've got some nice data to show, to talk about that a bit more. And then we've deleted ferritin in macrophages or myeloid cells, specifically using the lysine-CRE system. And we were lucky enough to have what we call a pseudo ARDS model in the lab, where we expose animals to high oxygen for a number of days to mimic ARDS. And we're seeing some really, really nice phenotypes with the ferritin lysine-CRE animals. We hope to actually submit this very, very soon. So we hope to be able to tell you about that very soon. And that's it. I want to thank all my collaborators and funders, especially Science Foundation Ireland is the reason I'm able to come back to Ireland, and the Irish Research Council for sponsorship of Lynn's PhD. And I also had a K99 from the NIH, which allowed me to get going. So I'm ever grateful for that. And that's it. Okay, I'd love to take any questions if you have them.
I have a question. Chris Chathamber, can you hear me? Hi,
Yeah.
hi. Lovely talk. Thoroughly enjoyed it. I'm in Milwaukee at the Medical College of Wisconsin.
Thank you.
My long involvement in iron and other metals. My question is, you know, in patients who are in the ICU, for example, their lung infiltrates, there are a lot of cytokines and such that are released. Have you looked at impact of cytokines on iron in the lung? Any studies pending?
Well, I know IL-6 is closely related to iron levels in the lung, but I don't know if anyone's actually given cytokines directly, like instilled cytokines into a mouse lung and looked at what effects it has on the microenvironment in terms of
that or chemokines,
iron biology. Yeah, I'm not aware of that.
Yeah, one wonders if there's a link there with the rage of cytokine storm and iron release and damage.
I imagine so. Yeah, the oxidative burst, I mean, surely requires some iron as well.
Okay, thank you.
Thank you.
Other questions? Michael.
It's more of a comment, but I'd like to draw her attention, Suzanne, Dr. Cloonan's attention to DMT1 as well. 2005, I had an opportunity for a sabbatical at the EPA.
With Andy Geo? Yeah.
With Andy Geo, yes.
I know that worked well.
Okay, yeah. And so he had me very excited. And of course, we were thinking then that it would be the plus IRE form of DMT1 that would respond to challenges. But it turned out it was the minus. The plus stayed stable in the cells and in the lung samples that he got from patients. And I found that very fascinating, although I can't interpret it. And that's why I'm going to ask you about thinking about in terms of any of your data. The second point is just a very recent one. We have found DMT1 also on the outer membrane of the mitochondria. And generally speaking, there's no strong evidence yet that it is the primary route for getting iron in, but it is a route for getting iron into the mitochondria. The mitopherins would take up action once the iron has passed the outer membrane. So again, it's just worth keeping it in your thoughts is the comment part. But the question is about DMT1. Do you have any data on it?
Yeah. So DMT1, I think, has come up in RNA-seq of human COPD macrophages. It's definitely there. It's always there, and I always see it. And it's something that I'd love to follow. And I know Andy Geo's work really well. You know, he was one of, probably I would call him a mentor early days, because he would give me a lot of feedback on my work, and
good.
Really encouraged me to stay in the field, because he had found it quite tough, and you know, obviously there was other things going on for him to continue. But his theory was that with smoke, that the iron changes were all down to this particle effect that he called. So basically, if you've got these tiny particles being accumulated in epithelial cells or in macrophages, the macrophage or the cell thinks that it's iron starved from whatever effects these particles are having, and so therefore it thinks it needs to take in more iron. And that's what his logic was. And I've never been able to really, it's something we get asked all the time, is where is the coming from and why.
I can spin that slightly differently for you.
Yeah.
Although, and I think Andy, at least I wouldn't disagree with this. In essence, he thinks that this is part of the innate immune system, and that if you sequester iron in the macrophage, not only does the role of ferroportin and not letting it go out, but there is the role of iron in keeping it in, and that's also true for the epithelial cells. So hiding the iron, because particles are often associated with being, or some of them being bacteria or other invasive things.
Yeah, good.
So
I never thought of it like that. Yeah.
That's how.
Pretty cool. Yeah. And then in terms of the DMT1 and the outer mitochondrial membrane, that's really interesting. What I will say is that when in the data I just showed you with the mitoferrin deficient animals, if we give iron back to those animals, you can restore the phenotype, they survive. So the iron is getting into the mitochondria some other way, right? But the mitoferrins are not there. So, and I think the field has sort of known this for a while, that mitoferrins may not be the only way that iron gets into the cell, but definitely they're very important if you deplete both of them at the same time. So if you give a high iron diet back to these mice, they survive, and so that suggests to me that it's getting in somehow. But we haven't looked at the role of DMT1 and in the mitochondrial transport, but we should.
It's hard because it's on the plasma membrane too, of course.
Yeah, it's hard to measure mitochondrial iron. It's really hard. When I first started in this field, I was like, why is nobody? There's nobody measured this in easily, and I was like, oh, because this is why. It's really hard to do.
We have another question in the chat. Carolyn Philpott, do you want to ask yours?
Yeah, sure. So sort of along these lines, hi, Suzanne, I enjoyed the presentation. It makes me think about our PCBP1 macrophages.
I know, we keep going back to that all the time. Bill brings that up all the time.
We should stay in touch.
Yes.
Why is there such a strong inflammatory response in your mitoferrin 1 or your mitoferrin 1 and 2 double knockouts in the alveolar cells? Why is that?
Well, I think the cells are dying in the alveoli. So I'd say what's happening is the type 2 cells are just dying, and then that induces all this injury coming in to try to get rid of it.
Okay, so even though there's still uptake of iron into the mitochondria, these cells are just dying.
Well, yeah, I think so. I think if you delete both, I don't know if. So we haven't been able to measure mitochondrial iron specifically in isolated type 2 cells from the mice, simply because you can't get enough starting material to make mitoblasts. It's, and as far as I'm aware, there's no probe, and Bob Hyder's stuff, he in vivo, we can't do it, right? We can't know for sure. And I haven't measured death marker. We haven't looked at death yet. So I can't absolutely say that they are dying. I just know that if you give the iron, you kind of restore their survival, and I don't know why that is. I really don't. And maybe we give the iron in the diets as we induce the deletion of the genes. So something we may want to do is maybe give it later, afterwards, and see can we repair or restore the animals rather than circumvent the death. But I don't know if the iron is still getting in or not in those animals. We can maybe address this in vitro a bit better.
We have another question in the chat. Go ahead and ask it. I'm gonna miss, is it Ciara?
Kira Shaver from Vanderbilt in Nashville.
Hey, Kira.
Hi, that was really beautiful, Suzanne. I always love hearing you speak.
Thanks.
Can you comment on what you like? Why is iron handling different in the airspace compared to the circulation? Do you think that is because the airspace is generally iron deficient in a way that's different from the circulation?
Yeah, well, I mean, the oxygen tension is different, right? So I think because it's so sensitive to oxygen, and I think that's one reason why it's different. And I think the second reason is because of the microenvironment is so lipid rich. And we all know the role that iron and lipids play together. There's a lot of papers in ferroptosis in these models now, maybe that the iron is limited to inhibit ferroptosis, who knows, for lipid peroxidation, but definitely it's different because of those two reasons. I really think the oxygen and the lipids are unique to the lung.
Fair enough. It's awesome. I'm looking forward to seeing some of the work you.
And congrats on your R1. Did you get an R1?
Thank you very much. Thank you.
Nick Bessman has a question.
Yeah, thanks, Diane, and thanks, Suze. That was really another great talk. I'm just curious, have you guys looked at ex-smokers, humans or mice? Like, do their macrophages kind of normalize rapidly?
We haven't looked at it, but I mean, it's well known that if you've smoked for many years, you have baseline inflammation for a long time. In terms of time from smoking cessation and iron staining, we have former smokers in our studies, but we haven't compared them easily. Like, we haven't looked, I don't think anyone's looked at pearl stain macrophages of those patients. I imagine it starts to resolve, but I wouldn't be able to comment or confirm that for sure.
Okay, great, sure. I was just curious, thanks.
Okay.
So I had one question myself. The macrophages that you showed early on in your talk look like the iron is accumulated in vesicular compartments.
Yeah.
So similar to the innate immune response, it looks like they're sequestering the iron into those compartments so that pathogens aren't getting it. Have you looked at the levels of the reductases that are present on those vesicles? Because there's a couple of published papers, one which is mine more recently, where you actually, in response to pathogens extracellularly through a MIghty88 TLR4 dependent pathway, downregulate those reductases to retain the iron in those vesicular structures. So maybe in your smokers model, you're downregulating those reductases, which are necessary to convert the iron to be able to transport it through DMT1.
I haven't looked at it, but I definitely will now.
Yeah,
and something that Bill keeps going back to is in these stains you can see these punctate features, both in the human and the mouse. And what is it? Is it lysosomal? Is it what is it? We don't know. So that's something that he wants to do by imaging. Again, it's hard, as you guys all know, it's hard to image the iron very well in these sort of situations. So
so take a look at STEEP 3, but also sorry for the nomenclature, not mine, CYB 561A3, which is another reductase. We call it L-site B, but it's found on the lysosome.
Yeah, I imagine it is probably altered by smoke. Many of these type of enzymes are, because there's so many chemicals in the smoke, and there's the downstream metabolic processes as a result of smoke are pretty wide.
And so your tuberculosis model ought to be super happy. Yeah, it ought to proliferate quite well. Does anybody else have a question they want to ask? You're welcome to unmute yourself and ask it or make a comment. Okay, I think that's it. Thank you again, Suzanne, for the presentation. Super fun. I'm looking forward to knowing there's a whole pile of publications that many of you have that are sort of backed up, and hopefully will come out in the next year so we can continue to learn more and continue these virtual seminar series to keep connected when travel is complicated.
Thank you so much for the opportunity, and maybe see you at the European Iron meeting in July, if any of you are going to take the trip.
Okay, great. Thanks so
Diane.
much, guys. Take care, everybody.
Bye. Bye.